Hello, and thank you for connecting to this presentation. My name is Tom Farrell, and I'm here to tell you about Artiva Biotherapeutics, where our mission is to make cancer cell therapy accessible to any patient who stands to benefit. Our platform and pipeline are built on natural killer, or NK cells, which have risen to prominence in 2020 thanks to compelling clinical proof of concept data. NK cells have the potential to be delivered off the shelf, but manufacturability challenges have held the field back. At Artiva, manufacturing is job one, and we have a fully realized proprietary NK manufacturing platform that successfully addresses these challenges. We have a seasoned cell therapy leadership team and a dual product strategy that encompasses monoclonal antibody therapy on the one hand, leveraging the natural biology of NK cells as mediators of ADCC, and armored CAR-NK candidates on the other that fully exploit the recent clinical POC data. We also have exclusive access to a CRISPR platform that we can use for persistence enhancement or knockout of inhibitory pathways as needed, completing our toolkit. We raised a Series A financing in June 2020 from 5AM Venbio, RA, and a group of Korean investors, including our corporate partners, Green Cross or GC. These funds are going to be used to establish clinical proof of concept for 3 product candidates. AB101 in combination with monoclonal antibody therapy, specifically rituximab, in aggressive and indolent NHL. AB201, our first CAR-NK targeting HER2 in breast, gastric, and bladder cancers. And AB202, an armored CD19 CAR-NK that will be advancing in B-cell malignancies. This is just a snapshot of the data that I alluded to earlier from MD Anderson and Katie Rizvani specifically, and many of you are probably familiar with this, published in February. The product is an allogeneic cord blood-derived CD19 CAR-NK product that co-expresses soluble IL-15. This is now partnered with Takeda. They have a limited scale of production, and the product is not cryopreserved. In other words, it's given fresh and is not a true off-the-shelf product in this embodiment. Most impressively, the efficacy here is comparable to what's seen with the approved CD19 CAR T-cell therapies. Durability of anti-tumor activity is confounded by post-treatment therapy, and there was also no ability in this setting to repeat dose. The great news here is from a safety standpoint, no CRS or neurological toxicities, no graft versus host disease, which is as expected for an NK cell, and no tumor lysis syndrome, and no grade 3 or above non-hematologic toxicities. So NK cells then have terrific potential from an efficacy standpoint. Safety, as I alluded to, there is no T-cell receptor, so no risk of graft versus host disease, which fundamentally allows off-the-shelf allogeneic use. And they do not release pro-inflammatory cytokines, which reduces the CRS and neurological toxicity risk. It's a versatile platform as well. They're a natural mediator of ADCC, and that can therefore be combined with antibodies that work through that mechanism of action, or they can be engineered with CARs, etc. The challenges I've already mentioned, NK cells are fragile, they're hard to work with, hard to scale, hard to successfully cryopreserve, engineer, and edit, which is where our platform comes in. So we have a process which I will walk you through that gives us very large scale expansion, maturation, and activation of naive starting NK cells from cord blood. We can cryopreserve our final product, which is essential for patient access and repeat dosing, and a low cost of goods. All of this allows a high quality product that's manufactured on a campaign basis from healthy, selected starting cells with a robust process that gives us excellent purity, viability, and consistency. So with our platform applied to primary allogeneic NK cells, we can deliver large-scale, high-quality, cryopreserved, and infusion-ready product candidates to patients. The company was founded last year to develop and commercialize NK cell therapies outside of Asia Pacific. We're in San Diego. The team includes myself, where my background includes 11 years as CEO of Bellicum Pharmaceuticals. Prior to that, founder and CEO of Svilene Pharmaceuticals, a small molecule drug discovery and development company. So I've been in this for about 20 years. Pete Flynn spent 7 years at Fate Therapeutics as head of early development, where he led the development of Fate's iPSC platform. Jason Litton joins us from Juno Therapeutics, where he was head of solid tumor opportunities. And then the senior leadership team rounded out by Gene Helsel, head of regulatory and quality, and John Lim, head of clinical operations. I should talk about our corporate collaboration with GC Lab Cell. So GC Lab Cell is a global NK cell R&D leader for the past decade that is based in Seoul, South Korea. They're operating out of a brand new building, 300,000 square feet of research, process development, and commercial ready GMP manufacturing in the building there on the left that's haloed in red. And through an exclusive relationship with them, we have ex-Asia Pacific rights to their core IP and options to exclusive licenses for product candidates that emerge from their research engine. So let me spend a couple of slides walking you through the manufacturing process. So this is a proprietary e-feeder-based system that we're applying to allogeneic cord blood-derived NK cells. We start with licensed cords that are sourced here in the US. We're working specifically with the New York Blood Center, which is one of 8 licensed public cord banks in the US. Cords are selected based on 2 favorable attributes that are important for our final product, and then shipped cryopreserved to the manufacturing facility in Seoul, where they are expanded and activated over a process that's about 4 weeks. It's 2 phases. In the first phase, we isolate mononuclear cells. So this is not a differentiation approach. So we expand those cells to a master cell bank, where we generate typically around 80 units from each starting cord, and those are then cryopreserved. We thaw an individual vial and use that to seed a 50-liter bioreactor step, which then gives us approximately 80 to 100 vials of 1 billion cells per vial of final drug product. That again is cryopreserved, can be shipped to a 3PL, currently here in the US, from where we distribute to our clinical trial sites. So this is truly a remarkable scale of production. It allows us to make between 4,000 and 8,000 vials of drug product that can treat between hundreds and thousands of patients, depending on the dose level and the number of repeat doses that we intend for each patient. For an engineered product candidate, we add a lentiviral transduction step ahead of the master cell bank and then cryopreserve at that point. So what about the quality of this process? So on the left, you're looking at the phenotype of 3 starting cord units. And what you can see there is a high degree of consistency, but a relatively immature phenotype. As we move through the process and get to our final drug product in the top right, you can see that these cells have now been activated. They're expressing important engagement markers on the surface of the NK cells, such as NKG2D or NKP30. And they're also expressing very high levels of CD16, which is vital for engaging the Fc region of the antibodies that we're looking to partner with. Bottom right, you can see that we also have very consistent expansion, high purity, and highly potent cells at the end of this process. So moving on to our product pipeline, and this is where you can see this bifurcation in our strategy, where on the left-hand side, we're looking at monoclonal antibody enhancement. So product candidates that are broadly partnerable, universal products that are paired with either ADCC or CPI monoclonal antibodies or with novel binders. Our product candidate, and our lead product candidate here is AB101, is an ideal phenotype with high expression of high affinity CD16 and activating receptors. Our second focus, proprietary armored CAR-NKs, includes NK-specific co-stimulatory domains. So we have not simply taken CAR T-cell co-stims and dropped them into an NK cell. We express soluble IL-15 for enhanced persistence. We have high efficiency lentiviral transduction. And importantly, we maintain the high affinity CD16 receptor, which gives us the opportunity to combine our CAR-NKs with antibodies, as well as our AB101 product candidate. So our product candidates then include AB101 on the left, where our first combination is with rituximab in relapsed refractory non-Hodgkin's lymphoma, and we expect to start clinical trials with this program this year in the fourth quarter. Our first CAR-NK, AB201, is targeted to HER2. We're currently evaluating potential antibody combinations to include at some point in the development of this assay. In our initial clinical program, we're going to be targeting HER2-positive gastric, breast, and bladder cancers, and we expect to file our IND next year. Lastly, AB202 is our CD19-targeted CAR-NK, again, with antibody combination to be determined, with indications, as you would expect, including B-cell malignancies and an IND towards the end of 2021. So that concludes the presentation. So again, NK cell therapies are poised to break through in 2020 and beyond. We have a manufacturing platform that solves the central challenges that have dogged the NK cell space over the past decade or so. A compelling product pipeline that will be moving into the clinic in the near term, and hoping to be able to report on clinical data in 2021 and beyond. So thank you again for your attention and listening to this presentation. And obviously, I'm happy to receive any follow-up at Artiva, and I've listed our website here on the final page. Thank you again for your attention.